All right, so this is chapter 9, part 1. It also has a little bit of part 2 of it. So it's a little bit of a longer presentation. All right, so again, we're in chapter 9. Here are all the objectives for the whole chapter. You can read through those. Those are what you should be able to do at the end of this chapter. A lot of vocabulary. Some of it we've already talked about in lab. So in this chapter, you're going to learn about Gregor Mendel, genetic crosses, the law of inheritance, extensions of the law and inheritance, chromosomal basis of inheritance, and patterns of inheritance. So we start with Gregor Mendel. He joined the Order of St. Augustine and lived in St. Thomas Abbey in Brno, which is now the Czech Republic. He worked in the Abbey's garden between 1856 and 1863. He actually was the son of a wealthy physician, actually a family of physicians. His grandfather was also I would say an evolutionist as well. He studied embryology. So there was a history of research in that topic. Gregor Mendel did not do well, and he went to med school and he kind of failed out of med school. And my understanding is he couldn't even stand the sight of blood. So he decided to go into natural sciences. And so with that, he then ended up joining this order where he could carry out his research. And he is commonly known as the father of genetics. So Mendel worked with various different organisms, but he found his peas worked the best. And so he's famous for his peas. And a characteristic is a physical feature of an organism. For example, human height, hair color, eye color, those are all characteristics. But in peas, he looked at stem length, whether they were tall or short. Where is the flowers? Are they at the end of the stems or in the middle? The seed texture, are they wrinkled or smooth? The color, the pod texture, the pod color. So all of these things were easily identified characteristics, or we can call it a trait, which is defined as the variation of that physical form of that characteristic. Is it tall? Is it? Characteristic would be height. The trait would be tall or short. So here's a picture of those different things that he was looking at. So Mendel worked with these traits, and he showed that we have what's called discontinuous variation. Discontinuous variation is when each individual exhibits one of two easily distinguishable traits. Versus continuous variation is when a population displays a wide range of values. So like, for example, what we're saying is like, you're either tall or short. Versus in humans, well, you could be tall, short, or in between. So there's this continuous variation, like skin color. There's a continuous different colors versus saying, well, you are either white or black, which peas are either green or yellow. So that's the difference between discontinuous and continuous. Because of continuous variation, many scientists of Mendel's day subscribed to the blending theory, thinking that it kind of makes sense that if you have someone with dark skin and someone with light skin, their children are often in the middle, which is a blend of the parents. And what we have found is that's not true with Mendel. Skin color is kind of true because of a topic we'll talk about, incomplete dominance and multiple genes a little bit later. But the things that Mendel looked at was he looked at these discontinuous variations, these very clear cut forms of a gene: purple flowers, white flowers, green peas, yellow peas. So very simple characteristics he looked at. We know with these species, flowers can self-fertilize themselves. They can either self-fertilize or cross-fertilize, or we call it self-pollination or cross-pollination. And again, that's when the pollen, which many flowers have both males and female parts. So that's how come they can fertilize each other. And so he did this self fertilization over and over and over until you got true breeding pea plants or purebred pea plants. And what we're saying is that every time he crossed those plants, the offspring looked just like the parents. Mendel performed hybridizations or cross-fertilization with two true breeding mates. So we tend to call the true breeding the beginning, the first generation, our parentals or P generation. And then the hybrids or the first generation from the parentals as the F1 for filial, which means son. And then he crossed the F1s together to get the F2 generation. So here we have a purebred purple and a purebred, where did my pen go? A purebred purple and a purebred white. Okay. And so those are our parentals. When he crossed the F1s, we saw purple. So now we know that purple is the dominant trait. It's covering up the white. And then he crossed those with themselves. So we could sit there and say, well, we had purple, big P, and little p, little p. And so this is big P, little p. And now we crossed that with each other. And we ended up seeing the white back. I was like, oh, well, if this was blending theory, if this was truly blending, we would have gotten like a light purple. And then light purple with light purple, they would all be light purple. And they'd all start looking the same, which is not what we find. So we did many of these crosses. He counted many P's, and we can see the ratios are what we call 3-to-1 of our F1s, which is what we would expect. So Mendel's conclusions are that each plants carry two copies of an unchanging factor he called genes factors to determine characteristics. Because there are different traits for the same characteristic, there must be different versions of each gene, and he called these different versions alleles. Individuals that have two identical alleles are homozygous. Individuals with two different alleles are heterozygous. So Mendel's conclusions were that Mendel suspected each parent passed only one of its alleles to its offspring. The male and female gamete would carry only one copy. And we talked about this during meiosis, and that would be our divorce. So when we fertilize, we get back those one copy from mom, one copy from dad. So our zygote is back to being diploid. Mendel proposed what we call the law of segregation, which basically means that those homologous chromosomes separate during gamete formation. And so this is what, again, this is basically the technical word for that divorce. We're segregating, we're moving apart our homologous chromosomes. Because the F1 plants carry two different alleles for the genes, but only express one trait, one allele is dominant, the other one is recessive. The allele is considered dominant when it expresses, when we see that characteristic in the heterozygous. So like I said, the violet is the dominant for P flower color, because when we cross the purple with the white, we see purple. It masks or covers the recessive trait. When we talk about our genotype and our phenotype, so Mendel could only observe an organism's actual physical traits. So what we actually are seeing is called the phenotype. The genes that they carry are the genotype. So when we're going to represent genes with letters, so if we look down below, we have our big B, which is our genotype for our homozygous dominant, which gives us a phenotype of a purple flower. Our big B, little b is heterozygous, and that also gives us the phenotype of a purple flower because the big B is dominant over the little b. The homozygous recessive little b was the only way we can see the recessive is to be homozygous, and now our flower is white. So we have what's called a monohybrid cross. The monohybrid cross is also preferred. We're going to use a tool called a Punnett square. And so when we fertilize our eggs and sperm, we can figure out the probability of what our offspring should be by using what's called a Punnett square. So for a monohybrid cross, we're going to have a 2x2 table. The alleles from one parent goes on the top, the alleles from the other parent goes on the side, and then we're going to put them together in the body of that table to get back our diploid offspring. So we can see here where we have our dominant, our homozygous dominant, our homozygous recessive. When we put it together, all of our F1s are going to be heterozygous. When we take our heterozygous, so we're saying, well, in some of the eggs, we'll have a big Y, some of the eggs will have a little y. And so when we cross those together, we have, so I'm just going to do that here in my cross. So we started with big Y, little y, big Y, little y. Our other plant was also big Y, little y. So you're going to move the big Y across and the little y across. I purposely put the little y off to the side a little bit because we like to put our dominant letter first. Okay. So what this is giving us is our GER, our genotypic ratio, is basically saying, well, what genes do I have? Well, I have big Y right here. I also have big Y, little y right here and right here. And I also have little y, which is right here. That is just the possible genotypes I have. But how many do I have? Because I want a ratio. Well, I have one square that's big Y, two squares that are big Y, little y are heterozygous, and one square that is little y. So my ratio is a 1:1. Now, when I look at my PER, this one here is going to be yellow, 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 green. So my ratio will be 3. You always start with the dominant. 3 dominant to 1 recessive, or 3 yellow to 1 green. All right. And that's what we're seeing here. We get that ratio of 3:1. Okay. So that is it for the first part of chapter 1. So you should be able to do what's called the monohybrid cross. I have practice stuff for you, or a single cross. And that means we're looking at just one trait at a time. And we're going to move up in difficulty once you have that mastered to a dihybrid cross, which is 2 characteristics at a time. I hope this is helpful, and I will see you in the next video.